Hi class, Dr. Jim here. In this lecture we're going to be looking at what everyone's been waiting for all semester, I know, and that's building Punnett squares and putting together monohybrid crosses. And so I know I can feel that excitement in the air. I can feel it through the air, basically through this video, knowing how much excitement you have. And I think the real issue is that a lot of people struggle with these things, and the reason why they struggle is because they don't know how to set them up in the first place. So hopefully today what I'll be able to do is take you step by step on how to set one of these things up so that you feel really comfortable in answering the questions that we have. And again, practice makes perfect. I left you a ton of different problems on Blackboard for you guys to actually do so that you can get really comfortable and master the monohybrid cross. There are also two other lectures that kind of accompany this lecture, and the reason why I separate them out is so that you kind of get one mastered first and then look at the other two after the fact. The other two, the first one is kind of the special situations. What happens with the monohybrid when we talk about special situations, like when you talk about incomplete dominance or codominance or even these X-linked traits. And we'll kind of look at those in another lecture. And then the other thing that we're interested in is putting together a dihybrid cross. But if you don't understand the monohybrid, or that's really confusing to how you even put one together, then don't even worry about the dihybrid. Figure out how to do the monohybrid first and then move on to the dihybrid, okay? So that's why I kind of separated out in 3 distinct videos so that you kind of work with your comfort level, get better at one, and then move on to the next one and to the next one. It's kind of like going from beginner, intermediate, and advanced, okay? So that's kind of how I have it set up. So let's begin with the monohybrid Punnett squares. Okay, and so today what we're going to look at is the terms that we use. And so this is just a basic looking at some of the terms that are utilized throughout a lot of these problems. And I want to make sure that you understand what these terms actually mean. So it makes it much easier to understand how to do these Punnett squares when you understand what all the terminology means in the problem itself. The other thing I want to do is show you how to perform and actually do a monohybrid Punnett square. And at the very end, I'll actually have a problem for you to actually work on. You can pause the video and then I'll show you the answer to kind of give you a little more comfort and a little bit more encouragement to actually being able to do these things. All right, so let's kind of look and see where we're at. So what are some of the terms that are used in genetics? Well, probably one of the most important ones is allele. An allele is basically used or interchanged a lot of times with both the word gene and trait. But it really shouldn't be, because allele is kind of something that's different. Allele is described as a version of a gene. Okay, let's say in this make-believe world where we only have one gene controlling our color, our height, our eye color, things like this, we can say that we have 2 different versions of everything. We either, for our height, we're either tall or we're short. There's no in between. Okay, or for our eye color, we're either brown-eyed or blue-eyed. We don't worry about the hazel eyes or the green eyes or other things like that. It's just one or the other. Okay, and when we talk about in a simplistic term what an allele is, it means that you have 2 different versions of that one gene. Okay, you're either brown-eyed or you're blue-eyed. Okay, and so that's what an allele means. It's a different version. And again, you get one from mom and you get one from dad. And depending on what they have, that determines what you're going to get. Okay, and we'll look at all these things during the Punnett square and kind of give you an idea of how you pass genes along and where we get those from. Okay, the second definition that I really want you to know is genotype. Genotype refers to the letters of the alleles. Okay, and what we mean by that is that the letters describe the version that you have. A lot of times they're either capitalized or lowercase. The capital describes the dominant or the one you see more of, and the lowercase describes the one you see less of. All right, and so that's going to be one of those terms that we use throughout this whole lecture. Phenotype describes the appearance that you see in people. So this would be, you know, if we're talking about blue-eyed versus brown-eyed or tall versus short, this is what you actually see with your physical eyes. Okay, and so again, very simplistic form, either one version or the other. There's no in between in these types of problems. All right, and then we talk about dominant versus recessive. Dominant means this is the one you see more of, and so you can actually look at these things and kind of figure it out based on what you see. And if you remember back to the Gregor Mendel lecture that we had last time, what he did was he looked at 2 traits. He looked at like tall versus short, green versus yellow, and he noticed that one had more than the other. And that was the dominant versus the recessive. And so that's what we see here. And typically we describe the recessive as something that gets hidden by the dominant. If the dominant is ever present in your genotype, you're going to see the dominant trait. if the only time you see the recessive trait is going to be when it's by itself, when you have two copies of the recessive gene or the recessive version of the allele. Okay, so that's what we mean by either dominant versus recessive. And then we also talk about homozygous versus heterozygous. Homozygous means you have the same two versions. Either you have both tall or you have both short. Okay, again, no mix. Heterozygous is when you have one of each. So you have a tall allele and you have a short allele, or you have a brown-eyed allele and you have a blue-eyed allele. That's the heterozygous. Homozygous, the same. You have both brown-eye or you have both blue-eye. Okay, and so we'll use these interchangeably throughout, so make sure that you kind of get an idea of what these things are. And so I definitely would write these down and make sure that you understand what each one of these terms mean, because you're going to see them used throughout these problems, and so understanding this will actually help you put together these monohybrid crosses. So these are some of the terms that are actually used in the problems, and again, I think these are important terms that you should know. I have three real important ones that they always throw in these problems, and if you understand what they are, it's really kind of getting at what you need to figure out. Okay, so what is a monohybrid cross? Well, that's basically looking at one type of gene. Okay, so we might be looking at the height gene, or we might be looking at the color gene. Well, what are we looking at? We're looking at two parents and having that gene and what versions they have and what versions do they give to their kids. And so that's what the monohybrid cross is looking at. One gene looking at the different versions that the parents might have. Okay. The genotypic ratio is just concerning with the letters. And so typically when we look at the genotypic ratio, we're just concerned with how many big T, big T, big T, little t, or whatever letter you use in those things. The genotype is always to the letter. And typically when you're talking about the genotypic ratio, you always have three numbers associated with it, because you always have a group of big T, a big T, and a little t, or whatever letter you use, okay? And so kind of think of that genotype, I need three numbers. So it'll be like 3 to 2 to 1 or something like that, okay, depending on what you have. A phenotype is just looking at whether you're tall or short. So it's actually what you see, okay? And typically there's only two numbers associated with that in a monohybrid. So what we talk about in the phenotypic ratio is that you see tall and how many short, or how many blue-eyed versus how many brown-eyed, or how many green versus yellow. And that's what the phenotypic ratio represents, okay? And so the reason why I put these terms up there is you see these a lot of times used in problems, and if you understand them, then they're not scary terms, and you understand what they're actually asking in these problems, or what do you need to solve for, okay? So make sure you understand those as well, because you'll see those in the problems. Now, who do we get to thank for these wonderful Punnett squares? Well, the person that came up with these is Reginald Punnett, and this is a picture of him. He was a British geneticist, and he actually came up with these Punnett squares as a way to figure out what are genotypes and phenotypes of offspring. So we could see these before we even knew anything about DNA. We knew we passed traits on to our offspring, and this was one way that we could actually figure out what our offspring were going to look like. And we could utilize these things without even knowing what the DNA was by just looking at the parents' traits. Okay, so looking at the parents and what they have, kind of like Mendel and his pea plants. How did he know what were going to be tall and what were going to be short? Well, he could use these Punnett squares to kind of put these together. And Punnett was really the one that created these squares to utilize to figure these out. Okay, so what does the Punnett square actually mean? Well, you either look at the side or the top, and these represent the parents. Now, if you think about it, these are actually the meiosis, or the gametes that the parent produces. You remember, you can only pass one trait. Remember meiosis, you divide your DNA from going from 2 to 1 or 4 to 2 or whatever, how many traits you're talking about. If we're talking about one gene, you remember you have two copies of every gene. You can only pass one allele to your offspring. And so that's what happens in the meiosis. One allele goes here, one allele goes here. And same with the other parent. One allele goes here, and the other one goes here. What the boxes represent is fertilization. This is the fertilization of the offspring, so that you figure out what is the genotypes and what are the phenotypes of what your babies are going to look like. Okay? And in here, when we talk about fertilization, remember 1 and 1, sperm and egg, bring back and you get 2. So you always have 2 alleles inside these boxes. So you'll have 2 here, 2 here, 2 here, and 2 here. And so this will make a little more sense once we finally do a problem. And the next step we're going to do is actually look at a fun little one that involves me and my wife. Okay? So with the monohybrid cross, I have blue eyes. You might not be able to see them in this video, but the next time you look at me in class, you can see that I have kind of a bluish gray eyes. Okay? My wife, who you've never seen, who's not at class, my wife actually has brown eyes. And so we can actually use this, again, very simplistic model, brown eyes versus blue, and what are our kids going to look like? And so we ask the first question, what is our phenotypes? Well, that's pretty easy because I just told you. I have blue eyes. That's my phenotype, blue eyes. And my wife has brown eyes. All right, so that's how we figure out those pretty easy, blue eyes versus brown eyes. The next question we're going to ask is, what is our genotypes? Our genotypes are the letters or the alleles that represent our phenotype. So remember, blue eyes can only be seen when we have the two lowercase alleles, okay? So you could pick any letter, but typically when we talk about brown eyes versus blue, we use the letter B. We can use any letter that we want, or you can use any letter. Just keep them consistent. So the only time you can see blue eyes with the phenotype is when you have two small letters. So I know my genotype is going to be little b, because I use the B for brown, and the little b's represent blue eyes. So the only time you're going to see blue eyes is when you have little b, little b. The harder part is with my wife because she has brown eyes. Now, when you're brown eyes, you could have one of two different possibilities. You could either be big B, that's the dominant trait, you have brown eyes, or you could be big B, little b, that's the other trait you could be, or the other set or other type of allele that you could have, or combination of alleles. Okay, so how do you know which one to use? Well, typically in the problem they'll tell you, or they'll give you some clue to figure out what allele to use or what genotype to use for that parent. Otherwise, you have to figure out. Sometimes they give you their parents and you figure out from their parents to figure those things out. And I'll take you through step by step. But in this situation, let's just assume Amy has the big B. And so let's set up our cross. So we want to figure out what is our offspring's, both their genotype and their phenotype. Okay, so we put me on the side, and I typically put the male on the side and the female on the top. And what I would say is always be consistent. It doesn't matter if the male goes on top or the female goes on the top or whatever, but when you do these problems, be consistent. It just helps knowing where the parents are and what parent is what. Okay, so I always put the male on the side and the female on top, and here's Amy's genotype. And remember, even though we have two letters for our genotype, we can only give one to our offspring. So that's where these one letters go. This is the process of meiosis. You either give the little b or the little b, in my wife's case, the big B or the big B. Then we can look at her kids, and again, now you fertilize and you put two alleles back together. Remember, 1 and 1 goes back to 2. Okay? We can look. What are our kids going to look like? Well, the phenotype, what do they look like? Well, they're all going to have brown eyes. And what is their genotype? Well, they're all going to have one big B for my wife, and they're all going to have one little b for me. And so their genotypes are all going to be big B, little b, and their phenotype is going to be brown eyes. So if we look at the phenotypic ratio, we say brown eyes is 4 to blue eyes, which is 0. Okay, and what is the genotype? Well, the genotype is going to be, how many big B's do you see in this box? None. So that's 0. How many big B, little b's do you see? Well, you can count 1, 2, 3, and 4. There's 4. And how many little b's are there? Well, there's none. I don't see any little b's. So my ratio here is 0 to 4 to 0. And so remember, phenotypes typically 2 numbers, 4 to 0. Genotype ratios are always 3 numbers, 0 to 4 to 0. Okay, so I kind of give you an idea in that situation. So let's try it now where my wife has little b, or I'm sorry, big B, little b in that situation. Again, there's nothing in this problem that shows us, but let's just try this combination. Now you can see what happens is now we have a slight difference. Our phenotypes are different in our offspring. Instead of all being brown eyes, now half our kids could be brown eyes and half our kids could be blue eyes, depending on what allele my wife gives. Does she give the big B or the little b? So my wife is the important one in this case, okay? What allele does she give? So if we look, we can say our phenotypic ratio is 2 to 2. 2 children could be blue-eyed or brown-eyed, and 2 children could be blue-eyed. So we say 2 to 2. And what is the genotypic ratio? Well, again, we see no big B's in any of these boxes, so that's 0. We see 2 big B, little b's, okay, so 2. And we see 2 little b's over here, so that's 2. So our genotypic ratio is going to be 0 to 2 to 2, okay? Always 3 numbers. The phenotypic ratio is always 2, and that's 2 to 2 or 1 to 1 if you want to make it simplistic, but that's how we figure out the phenotype. So let's try a problem that you might see on a test, okay? So here's the problem. A brown-eyed man whose mother was blue-eyed marries a blue-eyed woman. What is the % chance that their children will have blue eyes? And what is the chance that they'll have brown eyes? So the first thing you need to do is figure out what is the phenotypes and genotypes of the parents. So let's do the woman because that's easy. She's blue-eyed, okay? She has blue eyes. She has the blue eye phenotype. We know her genotype because we know blue eyes is recessive. That's going to be little b, little b. Okay? We know the man is brown-eyed, but we don't know what his genotype is. It could be big B, or big B, little b. It's the same situation that we had with my wife, Amy. But how can we figure this out? Now, if you look at this problem, it gives you the clue right here. Mother was blue-eyed. So that suggests that the man had, this gives us some clue of what the man's genotype is going to be. So let's look at that. If mom had blue eyes, the only allele that she can give her son, which is the man, is going to be little b, because you can't give a big B if you only have little b's to give. And so we know that his genotype has to be big B, little b, because mom could only give him a little b. His dad obviously gave him the big B, and that's why he has brown eyes. Okay? So we know his genotype is going to be big B, little b. So you have to look in the problems themselves to see if you can find and see those things for yourself to answer those questions. So we put the mom on top, dad on the side, and we look at the possible combinations. And again, you just carry these letters over and you can kind of see how these things move. Okay, so again, big B, little b, those are going to be brown eyes, and then the little b, those are going to be blue eyes. So they ask, what is the percent chance that the children will have blue eyes? We now count 1, 2 out of 4, so that's 50%. Same thing with brown eyes. 2 out of 4, that's 50%. So that answers their question. So they didn't even want the ratio in this one, so you need to be careful and see what they're asking for. Sometimes they'll ask for the ratio, sometimes they'll ask for the percentage. And we try to be as clear as possible when we do these problems. So you need to make sure you understand. Are they asking for a percentage, or are they asking for a ratio when they answer these types of questions? Okay, so the next situation, I'm going to give you the problem. I'm going to have you guys pause the video, work on the problem, and see if you get the right answer. So let's do this problem. A yellow pea plant was mated with a homozygous dominant green plant. What is the percent chance the offspring will be yellow? And what is the percent chance that they'll be green? So here's the problem. Basically, I want you to figure out what is the phenotypes and genotypes of the parents, and then put it together and build the Punnett square. At this time, pause the video so you can do this before you see the answer. So pause now. All right, hopefully you paused and now you got the answers and that you're looking for. Let's take this step by step so see if you got this right. So the phenotype, let's take the easy one first, because the easy one is yellow, because we know when we see dominant green, that tells us that green is the dominant color, where yellow is the recessive color. Okay, so we have the recessive yellow, and we know again recessive is going to be the two little letters, right? Because when you see a recessive, you can only be little letters. Now the other one is asking, we know it's green, that's the phenotype, but how do we know what the genotype is? Well, we look again for the thing. Here's the key word, homozygous. Homozygous means you have the same versions, so you're going to have two of the big letters. If it was homozygous recessive, it would be the two little letters. So here you go, homozygous dominant. So we know that the genotype is going to be big G, big G. So we put this together, the little g, and the big G, and for plants it really doesn't matter which one's male or female, you could do either way. Again, just be consistent. And if you put this together, you carry the letters down, so the little g's go down, big G's go across, okay, and this would be your genotypes, and the phenotypes would be what they look like. And so when you have the heterozygous condition, this is always the dominant color. In this situation, the dominant color is green. So all four of these are going to be green, and all four are going to have big G, little g in the heterozygous condition. So when they ask what is the percent chance that they're going to be yellow, that would be 0 out of 4 or 0%. And if they're green, it would be 4 out of 4 or 100%. Hopefully you're able to get that right. If you weren't able to do that or set this up, I would go and look at a lot of the practice problems that I have. And don't be afraid to ask me for more problems. I'll be happy to give you a ton of problems to work on. And I really want you to get comfortable working on these things. If you can do these already, then now it's time to move on to the next video and watch about the special situations. So let's kind of review what we did. Okay? So to summarize, terms of genetics problems, you need to know the difference between dominant and recessive. Dominant is the dominant gene, one that's always shown. Recessive is the one that's hidden. Homozygous means that you have the same version of both genes. Heterozygous means you have the two different versions of it. Okay? Genotype refers to the letters associated with it. Phenotype refers to what you look like. Okay, monohybrid crosses, one gene at a time, looking at the different versions. Genotypic ratio means that you'll have 3 numbers associated. This is concerning with the letters, the big T, big T, big T, little t, little t, little t. Okay, phenotypic ratio is going to be always the appearance, and that typically has 2 numbers. Are you green or yellow, tall or short, blue eye, brown eye? Okay, always practice, 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 practice. Practice will make you help. If you don't have it yet, don't worry. Just keep practicing these problems. Once you really practice and get good at these things, you'll feel comfortable, and these will seem very easy to you, and then it'll be time to move on. Okay? Master the monohybrid before you move on to the dihybrid or the special situation so that you get a really good idea or a really good feel for these things, and so that you're really comfortable with it. If you have any questions, please ask me. Again, I'm always here for you. Please ask if you need help. All right? I'll see you next time, and thanks for watching.